Hello dear students, today I am going to speak on the topic mineral nutrition, methods of study and use of nutrient solutions, chelating agents. The main objectives of the present topic include: number 1, to understand the concept of mineral nutrition. Number 2, to categorize the mineral nutrients on the basis of their concentrations in plant tissues. Number 3, to describe various methods for the study of mineral nutrition. Number 4, to understand the role of nutrient solutions in sustaining rapid plant growth. And number 5, to know about the concept of chelation and the use of chelators. First of all, a brief introduction about mineral nutrition. The study of how plants obtain and use mineral nutrients for their growth and development is known as mineral nutrition. This field of study is important because mineral nutrients are involved in growth and other metabolic functions of plants. In addition, the study of mineral nutrition also helps in finding the ways to increase the production of modern agriculture to protect food crops and our environment. Plants are autotrophic organisms and live in an inorganic environment. They take carbon dioxide from the atmosphere and water and mineral nutrients from soil. Mineral nutrition is the study of how plants obtain and use mineral nutrients for their growth and other developmental activities. Only certain nutrients have been found to be essential for the growth and development of plants. These nutrients are known as essential nutrients based on the fact that a plant fails to grow and reproduce in their absence. In the presence of optimum sunlight and water, mineral nutrients help plants in the manufacture of all vital compounds. Essential mineral elements are usually classified as macronutrients or micronutrients according to their relative concentration in plant tissue. Macronutrients are present in concentrations of 1 mg per gram of dry weight of plant tissues, while as micronutrients are present in less than 1 mg per gram of dry weight. Macronutrients include following elements like nitrogen, potassium, calcium, magnesium, phosphorus, sulfur and silicon. Micronutrients include the elements like chlorine, iron, boron, manganese, sodium, zinc, copper, nickel and molybdenum. However, in some instances, the differences in tissue concentrations of macro and micronutrients are not significant. For example, leaf mesophyll of plants has been found to contain almost as much iron or manganese as they do sulfur or magnesium. It has been observed that some mineral elements have higher concentrations than the plant's minimum requirements. Study of inorganic nutrition is important because traditional organic nutrition failed to increase food production for the increasing world population. It has been observed that fertilization of agricultural fields with mineral nutrients is necessary for high yields. It has been observed that most of the commonly grown crop plants increase their production linearly with increase in mineral nutrient supply. The world consumption of commonly used mineral nutrients like nitrogen, phosphorus and potassium has increased tremendously from 112 million metric tons in 1980 to 143 million metric tons in 1990. The mineral nutrients constantly cycle through all organisms and enter the biosphere mainly through the root systems of plants. So, in a sense, plants are the mineral miners of Earth's rest. The effective absorption of mineral nutrients takes place due to higher surface area of roots and their ability to absorb nutrients at very low ion concentrations. The absorption of mineral nutrients is followed by their translocation to various parts where they are utilized in varied biological functions. Plant roots often form associations with mycorrhizal fungi. The fine hyphae of mycorrhizae extend the reach of roots into the surrounding soil and facilitate the acquisition of mineral elements, particularly those like phosphorus, that are relatively immobile in soil. In return, plants provide carbohydrates to mycorrhizae. Plants tend to suppress mycorrhizal associations under conditions of high nutrient availability. Now, let's discuss different methods of studying mineral nutrition. The methods for studying mineral nutrition usually involve the culture of plants in a soil-free medium. It's necessary that the mineral constituents of the medium are defined. These methods should be done carefully to avoid contamination either through water or mineral nutrients itself. Since a plant requires some specific elements, this demonstration is done by growing plants under experimental conditions where all nutrients except the required nutrient are present. However, it is difficult to create such conditions in a complex medium like soil. This problem has been solved in the 19th century by growing plants with their roots immersed in a nutrient solution containing only inorganic salts. This proved that plants can fulfill all their requirements from inorganic elements and sunlight. In other words, we can grow plants in absence of organic nutrients and soil. There are some analytical techniques to study composition of nutrient solutions. The two most important analytical techniques used are atomic absorption spectrometry or atomic emission spectrometry. These techniques work on a principle of absorption and emission of specific wavelengths of light by specific ions respectively. The amount of light absorbed or emitted is proportional to the concentration of the mineral nutrient in the sample. This helps us to calculate nutrient concentrations as low as 10 to 8 gram per ml in soil or a nutrient solution within a few minutes time. There are two ways of growing plants in absence of soil. These are number 1, solution culture or hydroponics. The technique of growing plants with their roots immersed in nutrient solution without soil is called solution culture or hydroponics. This technique was given by Julius von Sachs in 1860. To carry out successful hydroponic culture of plants, it is essential to have a large volume of culture solution. Furthermore, nutrient concentration of the medium should be frequently adjusted. This helps to prevent severe changes in nutrient concentrations and pH of the medium due to nutrient uptake by roots. A constant and vigorous shaking of the nutrient medium is essential in order to maintain a continuous supply of oxygen to the plants. A nutrient solution that is not aerated properly becomes depleted of oxygen. This depletion of oxygen is known as anoxia and inhibits the respiration of roots. Ultimately, further uptake of nutrients gets reduced. In some cases, the container containing the plants is wrapped with an opaque material or painted black. This blocks the entry of light and hence reduces the growth of algae which compete with the plants. Hydroponics is used in commercial production of money greenhouse crops. In some cases of commercial hydroponic culture of plants, Different types of supporting materials can be used. As an example, plants can be grown in a supporting material such as sand, gravel, vermiculite or expanded clay, that is kitty litter. This is followed by the flushing of the nutrient solutions through the supporting material and removal of the used solutions by leaching. One more way of growing plants hydroponically is to spread the nutrient solution on the surface of a trough in the form of a thin layer. The plant roots are kept on the surface of the trough with nutrient solution flowing in a thin layer across all the roots. This ensures a constant and ample supply of oxygen to the roots of the plants and hence favors their growth and development. The major problem associated with the use of nutrient solution is that of selective ion depletion by plants. This leads to pH change and poses major restriction to growth of plants. Also, plants in solution culture will grow if nutrients are added continuously. These problems can be avoided by the use of non-nutritive media such as acid washed quartz sand, perlite and vermiculite. Vermiculite is a silicate mineral of the mica family. It expands on heating to produce a lightweight substance which has a high water retention capacity. Perlite is a coarsely glassy volcanic rock. Both these substances provide virtually no nutrients for plants. Number 2. Aeroponics. This is another alternative for growing plants without soil and organic nutrients. It's considered as an important medium to grow a vast variety of plants in future. Here, plants are grown with their roots suspended in air and are constantly being sprayed with a nutrient solution. It has an advantage of quick and easy manipulation of the gaseous environment around the root. They also use little to no growth media. The plants also get more oxygen and hence show relatively fast growth compared to hydroponics. They also use less water than hydroponics and harvesting of plants is easier. This is particularly useful in case of root crops. However, there are some drawbacks associated with the use of aeroponics. It requires a higher level of nutrients to support plant growth than hydroponics. It is also expensive to build, and if there is any interruption in the watering cycle, the plants may die within minutes. Due to this reason and other technical difficulties, the use of aeroponics is not widespread. Now let's discuss uses of nutrient solutions in sustaining rapid plant growth. Many formulations have been used as nutrient solutions over the years to study the effect of nutrients on the growth of plants. Some of the early formulations developed in Germany by Noob included only potassium nitrate, calcium nitrate, potassium hydrate, phosphate, magnesium sulfate and an iron salt. This solution of nutrients was believed to have all the minerals for the plant growth. However, these experiments were done by using chemicals which were contaminated with other elements. Some of the essential elements found as contaminants in these nutrient solutions were boron and molybdenum. Recently, some modern nutrient formulations were also prepared. One of the important modern nutrient formulations is known as modified Hoagland solution. It was named after Dennis R. Hoagland, who was actively involved in the preparation of modern mineral nutrition solutions in the United States. The modified Hoagland solution is found to contain almost all the mineral elements needed for rapid plant growth. The amount of these mineral elements is essentially fixed at the optimum possible levels. This helps plants in avoiding the toxicity symptoms or stress due to salinity. The use of nutrient solutions leads to increased concentration of nutrients by several orders of magnitude around the roots of these plants than those grown in the soil. For example, concentration of phosphorus in the soil solution is normally less than 0.06 ppm, but in nutrient solutions it is offered at 62 ppm. Such high levels of nutrients permit plants to grow vigorously as compared to plants which are grown in the normal soil. This is also advantageous because the nutrients remain abundant for extended time without any sign of depletion. On the other hand, some of the workers use less concentrated nutrient solutions. This can be achieved by diluting nutrient solutions several folds. But this needs a frequent replacement of the nutrient solutions to minimize fluctuations of nutrient concentration both in the medium and the plant tissue. The most important property of the modified Hoagland formulation is that nitrogen is supplied as both ammonium and nitrate. This helps to reduce the rapid increase in the pH of the medium, since both the cations and anions are supplied in the form of a balanced mixture. Fluctuations in the pH are usually observed if nitrogen is solely supplied as nitrate anion. It has also been observed that even if the pH of the medium remains neutral, most of the plants show better growth and development. This is due to access to both ammonium and nitrate absorption and assimilation. As a consequence to this, the two nitrogen forms maintain cation-anion balance within the plant and hence avoid pH alterations in the plant tissues. In spite of the above described benefits of the nutrient solutions for plants, it is difficult to maintain the availability of iron in sufficient amounts. Iron is usually supplied to nutrient solutions in the form of inorganic salts like FeSO4 or FeNO3. The problem is that iron precipitates in the form of iron hydroxide and hence becomes unavailable to plants. The problem is accelerated if phosphate salts are also present in the nutrient solution because it results in the formation of insoluble iron phosphate. Precipitation of the iron and its unavailability to the plants means that iron has to be added frequently at regular time intervals. This problem was addressed by adding iron together with compounds called chelators. Finally, let's discuss chelation and the role of chelating agents or chelators. The problem of precipitation of iron and other essential nutrients of the nutrient solutions can be addressed by addition of iron together with citric acid or tartaric acid. These compounds form soluble complexes with cations such as iron and calcium. In these complexes, the cations are held with the help of ionic forces instead of covalent bonds. Chelated cations are found to be physically more available to the plant. These substances, which form soluble complexes with cations such as iron and calcium, wherein the cation is held by ionic forces rather than by covalent bonds, are known as chelating agents or chelators, and the phenomenon is known as chelation. Thus, chelators are involved in holding and providing the iron to plants in ambient concentrations. In the recent times, modern nutrient solutions are provided with chemicals like ethylene diamine tetraacetic acid EDTA or diethylene triamine pentaacetic acid DTPA. These act as strong chelating agents as compared to either tartaric acid and citric acids. The strong chelating nature of these agents makes the availability of iron and some other essential nutrient plants even easier. However, the fate of chelation complex during the uptake of iron by plant roots is not clear. It is believed that iron may be released from the chelator at the surface of the root when it gets reduced from Fe3 positive to Fe2 positive. The metal chelator then diffuses back to the nutrient solution to react with another Fe3 positive ion and other metal ions. This process of chelation and removal of the metal surface continues to make easy the absorption of these hard to absorb metals. After the uptake of iron with the help of chelating agents, it is kept soluble inside plant cells with the help of organic chelating compounds. It has been observed that citric acid is involved in the chelation of iron in plant cells and its long distance transport to other parts of the plant through xylem. This was all about today's lecture. Thank you.